Welcome to Euclide Academy. In this video, we're going to solve the 2017 Biology Paper 2. This is a theory paper. You have about two sections in this paper, Section A and Section B, and you answer all the questions in Section A and you answer about 3 questions in Section B. So this was for internal candidates, that is for grade 12. So let's go ahead and check out this paper and revise the questions and answer the questions which are there. Question number 1. Answer all the questions in the spaces provided on the question paper. Figure 1.1 shows some specialized cells. So we've got specialized cells here, P, Q, and R, and name each of the cells above. So this probably question number 1 is on cells most of the times. So you have to make sure that you get these questions right, because they're kind of the ones which are the easiest, because the ones to get the marks from. So name each of the cells above. P, okay, we can literally say that with P we are dealing with the root hair cell. So we can actually say this is the root hair cell. And then Q, of course, this is a blood cell, and to be specific is a red blood cell. So you don't have to write initials, you can write the full red blood cells. R, so R, if you can see on R, you've got the nucleus there, and you've got the vacuole and the cell wall. So definitely this is a plant cell. And then you can see some black dots inside. So these black dots are what we call chloroplast, and it contains chlorophyll. So the part of a plant which actually carries out mostly has this green stuff, carries out photosynthesis, and the part of the plant that is a leaf. And when we go in the leaf, internal structure of a leaf, we find you've got the epidermis, the upper epidermis and the lower epidermis, and then we have the spongy layer, and then we have the palisade layer. This is where we find the palisade cell. So this will definitely be the palisade cell. So the best way to tell, we can tell by those little black dots in there. There are many. So this is question 1A. So now let's go to question 1B. So this is question 1B, and question 1B says state the functions of cells P and Q, which is P and Q. Okay, so what are the functions of cells P and Q? So P is the root hair cell. So what does the root hair cell what does it do? So with the root hair cell, its job is to absorb water. It absorbs water and mineral salt. So absorbs water and mineral salts. Okay, mineral salts from the cell. That's the function of the cell P, which is this one. Then the cell Q, which is a red blood cell, its function is to carry or transport or carry oxygen around the body. So carry oxygen or transport oxygen around the body. So this should give you the 1 mark. Explain the adaptations for cells P and Q to their functions. So the cells P and Q to their functions, how do they adapt their functions? So adaptation of cell P, what makes it give it advantage? If we look at cell P, okay, it has got the root-like structure. So when we look at cell has got root-like structure, which increases the surface area. So adaptations for cell P, which is root hair cell, so that's got a root-like structure which extend out of its body, okay, which increases the surface area, for easy absorption of water and minerals. And then we've got adaptation for Q. So Q is the red blood cells. Red blood cells do not, you can talk about, they do not have a nucleus. So no nucleus. which in turn also increases its capacity to carry oxygen around the body. So increases capacity to carry oxygen around the body. So you don't even have to explain the features. I think just mentioning them is enough. Adaptation of cell Q, no nucleus. If you want to, can explain, but I think for 1 mark should be enough. But anyway, don't leave anything to chance. You can put it there. Adaptation of cell P, okay, the root-like structure, which increases the surface area. I would put those options there. Where in the leaf is cell R located? Cell R, which is this one. I think I've just mentioned this. We just mentioned this in the beginning. We said, okay, this is in the root, the palisade layer, the leaf, the palisade layer, that is where we find R, because of palisade. So you can say the palisade layer, palisade layer. Okay, that is in the leaf. Okay, leaf palisade layer. That is where we find that cell. Question number 2, figure 2.0. So I've got this graph right here, which is figure 2.0, rate of reaction. So here on the y-axis shows rate of reaction, on the x-axis it shows the temperature in degrees Celsius. Okay, so you can see here when the rate of reaction, the temperature is 5, the rate is here. When the temperature is 20, it goes up here. When it's 30, it goes up there. When it's 35, it goes up there. And when it's 40, it starts dropping, 45, like that. Okay, so the question now is, what is an enzyme? So an enzyme is a biological catalyst. It's a biological catalyst that speeds up the rate of okay, well, we can literally say the biological catalyst made. So you look at 2 marks, maybe you have to give more for you to get the 2 marks. The biological catalyst made of proteins, made up of proteins that speed up the rate of a chemical reaction. So I may just skip through some words here and then you can fill in. So that speeds up chemical reactions. So those are enzymes. So biological catalysts made of proteins that speed up the rate of chemical reactions. Of course, you can continue saying without themselves being changed, chemically changed at the end of the reaction. So it's up to you. But at this point, I think it's enough. What is, again, never leave anything to chance. You can go ahead and do that, but again, you're racing against time. Question 2A2. What is the optimum temperature for the enzyme reaction shown in the graph in figure 2.0? So the optimum temperature is the best temperature where you have the highest rate of reaction for that particular enzyme. So that is above that temperature, the enzyme gets denatured. Okay, you can't go any above that. So you can actually see here we're going up, then you go to the highest. Okay, it should be somewhere right there, and it should be somewhere there. So we have 35. So at 35, that's where you're seeing this tip there. Okay, then at 36, 7, kind of for a short period of time remains the same, and then starts dropping after maybe 36, 37. So probably we can go for 35 degrees Celsius. Oh, I should even probably include up to 37 degrees Celsius. B1 says, at what temperature in figure 2.0 is the enzyme completely denatured? So when the enzyme is completely denatured, that means it's actually no longer functions. So we shouldn't see any rate of reaction. So when we go to this graph here, you can see here it's able to produce to operate. So we're seeing the rate of reaction, okay, up to when it comes and it's dropping, and then we can see that after 55 there, 56 going to 57, it's kind of completely denatured. The rate of reaction is off right there. So I'll go for at what temperature figure 2.0 is the enzyme completely denatured. Okay, you can say 57 degrees Celsius or 58, okay, or 60 degrees is completely denatured. Okay, so you can see here just at the end of the graph right there. Give a reason for your answer in B above. Okay, the reaction stops completely. The rate of reaction, or the reaction stops completely at the temperature you picked. Okay, say 57 degrees Celsius, or there is no further increase in the rate of reaction after this temperature. Okay, that will be a better option. So there is no further increase in the rate of reaction, no further increase in the rate of reaction after that temperature which you've picked. So that will be a reason. Explain why it is important to measure the body temperature when a person is sick. So on question C1, explain why it is important to measure the body temperature when a person is when they are sick. Why should you measure the body temperature? So there are certain treatments which would work best under maybe certain body conditions or body temperatures. So you have to analyze which treatment will be best for that particular, in that condition that the body is in, or that body temperature. So basically this is to analyze the best treatment, to analyze the best treatment for that particular, or maybe the effectiveness of the treatment, how effective that treatment will be, effectiveness of the treatment based on that body temperature, to analyze the best or effectiveness of a treatment. I would go with that. Z2 propose 2 ways the body temperature of a sick person can be brought down to normal. But how to bring a temperature back to normal? So this could be, you can stay in bed and rest by resting. So you your temperature is high and you are exerting more exercise or doing more things, so temperature can go higher. So you want to rest, you want the rate of reaction to kind of the metabolism in the body to drop, so you can rest, go to bed and rest. Okay, so you can go to bed and rest, go to bed and rest. The other one, you can take a cold shower. Okay, or we can talk about a cold shower. So I'll go with those. And also drinking water or staying hydrated. Stay hydrated or drinking water, enough water. So stay hydrated. Okay, so this is what I would go for. Figure 3. This is question number 3. Figure 3.0 shows the life cycle of a mosquito. So the life cycle of a mosquito identified to stage. So when you talk about the life cycle of a mosquito, we have stage 1 here, eggs in water, then larva in water. Stage 2, stage 3, we have pupa in water, and then we've got the adult, which is stage 4. Okay, so these are the 4 stages they've shown us of a mosquito. So identify 2 stages in figure 3 which are most suitable for eradicating malaria. So the 2 stages which are most suitable for eradicating malaria. The one stage that we all know is the adult stage, where what are the methods that we see? We see people using mosquito nets, sleeping under insecticide treated nets. So stage 4, or the adult stage, is effective using treated nets, insecticide treated nets, stage 4. And which other stage? The other stage is by clearing the mosquito. You clear mosquito breeding places, tall grass in stagnant water. So that talks about the stage number 1, which is eggs in water. So stage number 1, you clear all those stagnant streams. You even spray on this where you have streams in tall grass. If not, you clear them out. So I'll go for stage number 4 and stage number 1. So the pupa and the larva are kind of difficult to clear out, and there are several studies which are actually showing that the several stages actually in the larva itself, and the pupa itself can be really difficult to deal with. So I'll go with stage number 1 and stage number 4. So for each stage in A above, state how malaria may be eradicated for each stage identified in A. Okay, I think we've been mentioned this for how malaria may be eradicated for stage for stage number 4, adult. This prevents sleeping under insecticide treated net, will prevent the mosquito from transmitting they'll prevent the mosquito, which is the vector, from transmitting the pathogen from to humans, and they'll be killed by using insecticide treated nets. So they prevent transmission of pathogen to host, and by using insecticide treated nets. So that is how you can prevent how malaria can be eradicated in stage 4. Stage 1, this clears breeding ground for the mosquito, which is a vector. So stage 4 helps to eradicate or remove the breeding areas for mosquito, and as a result reduces the population of mosquito, or the mosquito child of vectors. So it helps reduce the population of mosquitoes or mosquito. So that is by eradicating or destroying the breeding places of the mosquito. So it helps eradicate or reduce the population of mosquito. So that is how it would help to eradicate malaria. So just 2 ways in which malaria can be controlled in the community. Okay, we can just basically talk about the same options we've mentioned again. It's just a way of phrasing them. I'll just mention something a bit different. So way number 1, I can say wearing clothes that cover the whole body, or most of the body, or cover the whole body. That would help. And number 2, I'll just put it right here. We can say by the use of mosquito repellents in the community. Community members can use mosquito repellents Mosquito repellents. And of course, can you also talk about the third point by clearing breeding grounds for mosquito. So clearing breeding grounds for mosquito. And 4, by killing adult mosquitoes, okay, through spraying, indoor spraying. Okay, so these are all the ways in which malaria can be prevented in the community. B and C part. The B part says, what term is given to the mosquito based on its role in the transmission of malaria pathogen? So malaria pathogen is called a vector. The term that we use is we use it as a vector. So these are, a vector does not cause the infection, but rather it transmits a pathogen that causes the infection. Okay, so the term given to the mosquito in this one is the vector, which carries the disease cause, so the pathogen. Okay, which brings us to question C1. Define a pathogen. So a pathogen is a disease causing, so a pathogen is a disease causing bacteria. It can also be a virus or virus. So that's a pathogen. So it's a disease causing bacteria or virus. Forgive my handwriting. Okay. Name the malaria causing pathogen. So the pathogen that causes malaria, actually, which is being carried by the mosquito, which is the vector, is called the Plasmodium. So if you don't need further help than this, we do have online lessons for you. You can simply send us a message on the number on your screen, and we'll respond to you on how you register, and we can go about helping you to prepare for the exam. We do have lessons Monday to Friday. So name the malaria causing pathogen, that is Plasmodium. Define pathogen. Okay, we did that. So okay, so we can go to the next question. Figure 4.0 shows the longitudinal section of a stem. So this is the longitudinal section of a stem, and we've got the bud here and the stem, and we've got STU, okay, which is the vascular bundle, and the leaf. Which letters in figure 4 correspond to the cambium, xylem, and the phloem? Okay, so unfortunately we'll try to look here and see which is the, but okay, if you check properly, you can see where one ends and where one starts and how far one is inside. Okay, so in plants, and when we look at the stem, we've got the dividing cells of the cambium. They give us the xylem cells and the phloem cell. So the innermost, the one which points almost inside there, it's really difficult to see, but as you can see, S goes like really inside right there. Okay, so as this is the dividing, we have the cells which divide the cambium, which give us the xylem, then the phloem are on the outside. So if you look, you probably have something like that, and then which is that. So we can have the is cut into two. So this, if this was the stem, okay, so the ones which like pointing inside, we have the xylems, okay, xylem, and the one to outside, we have the phloems, So the outmost here, U, I think to go for the phloem, to go for U. Z, xylem, it will be for T, and then cambium, okay, will be for S. Okay, so that is, we can do it. And then we got the next one, which says explain the functions of S and T. So this is question number 4. Question number 4 now, 4A2. explain the functions of S and T. So what is S and T? S and T, I think we've mentioned that. So S is the cambium, and T is we'll talk about the xylem. So the xylem we did talk about even in the we talked about the root hair cells. I wanted to say about the first question. So the functions of T we mentioned is to transport water and mineral salts. Transport water and mineral salts. Okay, that is from the roots to the stems. That's the function of T, which is the xylem, and then the function of S, which is the explain the and T in the plant. So we talked about AT, which transports water in mineral salts to the stem, which is the xylem, that is for T. And then we have got S, which is the cambium. So the cambium is responsible for secondary growth. So it's responsible for secondary growth by undergoing cell division. So it's responsible for secondary growth, which is the cambium, it by undergoing cell division. So that's it for the cambium, which letter corresponds to the structure which needs companion cell to function. So the structure which needs companion cell to function is the one which is just right towards the back of the tree, like towards the end, this side, which of course to my eye is U here, and then T is getting over the other side, which is the phloem. So phloem needs the companion cell, so it will be that is U, right? So it will be U. So U. Which letter in figure 4 corresponds to a structure in which substance can either move up or down? So the phloem also transports like food around the plant. Food like goes when the materials come from the soil, like water, mineral salts, they go up to the stem and the leaf. That is through the xylem. But through the phloem, once the manufactured food has been manufactured, it has to be transported around the plant. That means to the stem, to the leaves, back to the roots. So phloem carries out that. So here I'll also go for U, which corresponds to phloem. So which is the phloem, and that is a structure which when food can go up and down, transporting around the plant. So figure 4 corresponds to a structure in which substance can either move up or down. Okay, that is U again, which needs companion cell, U, the phloem. Okay, so basically to how I can see things in this diagram. So U, the phloem, and T, which is a little bit inside there, I'll go for the xylem, and S, the cambium. Question 5. Mrs. Mumba delivered, okay, they had to pick Mrs. Mumba of all the names. Mrs. Mumba delivered a baby girl in a certain hospital. Okay, so this Mrs. Mumba has blood group O, and the father to the girl has blood group AB. Mrs. Mumba sues the hospital for giving her a wrong baby whose blood group was O. Alright, so we are talking about Mrs. Mumba. Unfortunately, Mumba happens to be my name. So Mrs. Mumba delivered a baby girl in a certain hospital. Okay, so it happened that she got the baby, but the baby she got had blood group O. So Mrs. Mumba has blood group O, and the father to the girl has blood group AB. Alright, so we need to understand genetics. We need to understand about alleles, recessive ones and dominant ones. So the question says, explain using a genetic diagram why Mrs. Mumba would win or lose the case. So when it comes to inheritance of blood group inheritance, we have got three contrasting alleles for blood group, or three versions. Normally you have two, like for height you have short and tall, the allele which calls for shortness, and one tall. For color maybe you have two, but for blood we have got three alleles. So you've got the allele which calls for O, and the allele which calls for A, and the allele which calls for B. So these alleles, the A and B, okay, so I'm just gonna put it right here, that the A, or let me just put it right here, the A and the B, these are co-dominant. So we explain in class what co-dominant means. So these are co-dominant, they both show. And then the blood group O is recessive, or the blood group A and B are all dominant over this. So A and B are all dominant. They are all dominant over this one. Okay, to that one. All right, so let's explain what would happen. So they are saying the Mrs. Mumba has blood group O, so meaning the only way that could happen that she has blood group O is that she has got two alleles for O. She's homozygous for O, so that would mean, so I'm going to get rid of this. So that probably would mean that she has got, so we can say we are using a genetic diagram. So the parental phenotype, okay, so parental phenotype, or P1, here we can see she's got OO, and I'll start with the gametes. So it could be she has got this one right here, and these are the gametes. Okay, this is what she has. Then the husband, or the father of the girl, has blood group AB, so meaning the father has got A and another one in A. Okay, so that means you've got, if you've got A and these alleles, your blood group is AB, since they are co-dominant, and we explain more in class. Or that's the blood group. So what could be the possible combinations here? So the possible combinations could be, of course, this could mean this could combine with that, so this also could combine with that one, right? And this, when they cross, could also come with this, and this again, when they cross, could also come with this one right here. So we can see that the possibilities, the child you can have one, she could have a child who has got an allele for O and another one for A, so meaning the blood group here will be A, since A is dominant over O, and this one will be O, and this other one will be B, so meaning the blood group will be B, and this one can be O. So she's only contributing the O because that's what she have, and then that's what she has. And then here could also be A, O, and A, so the child could be A, so these are the genes A, and here could be O, and the child that would be could also come with B, so the child will be B. So what this is F1, which we call the first generation of the offspring. You can call it the F1, or first filial, according to what we did in class. I should think we understand this. This is the F1. So this will be blood group A, this child will have blood group A, this child blood group B. Since A and B are all dominant, this will be A and this will be B. So there's actually no chance that she will have a child who has got blood group O. In short, the probability is 0, so or 0% that she can have a child with blood group O. Hence, Mrs. Mumba will definitely win this case. Will win. She's right. There's no way she can have a child whose blood group is O, because her husband has got AB. Unless if her husband was also O, then probably yeah, that would have been the case. Her husband was AB. So let's go ahead, and the next question says, so the B part of the question says, what could have been the correct genotype of the actual father? So I presume the actual father of the baby who had a blood group O. So the actual should have had this. Okay, this would have been the genotype of the father for him to produce that baby. Or definitely if the mother had a blood group O, so that could have been. So you can put your answer below for B. So I think we are done with section A. I'm remaining section B of this paper, and we'll encourage you to learn more. You can join us for online tuitions, and we'll help you prepare for the exam, and we'll continue answering section B in class. And thank you for watching. So we'll see you in the next video. If you have any question, you can put it in the comments below. If any concern or contribution, put it in the comments below, and we'll see you in the next video.